Let's now begin with specification number 3. It focuses on describing and performing chemical tests for various biological molecules. In this section, we're going to understand how each test is carried out, what substances are used, and what kind of results or changes we should expect. To break it down, the iodine test helps us detect the presence of starch. Benedict's test is used to identify reducing sugars like glucose and maltose. The Biuret test is used to check for proteins. And finally, the emulsion test tells us whether lipids are present in a sample. We'll be covering each test one by one, looking at the procedure, the expected color changes, and the scientific principle behind them. Before we begin our lectures and practicals, it's important to go over some basic lab safety rules. Safety always comes first when working with chemicals and food samples. Always wear eye protection, especially when using substances like iodine, which can stain skin and clothing. Be careful with hot water to avoid burns, and handle all chemicals with care. If you have allergies, such as to peanuts, avoid using those samples. Your health is more important than any experiment. In our tests, you'll be working with different samples, and for solid foods, start by crushing them and mixing with warm water to prepare a testable solution or suspension. Each test has its own method depending on what you're checking for: starch, glucose, protein, or lipids. For lipids, the sample is crushed in ethanol, then filtered and mixed with water. We'll go through each practical individually, step by step,